approved two different tests under emergency use authorization category. So the two tests which have been approved by FDA, I'll be discussing it briefly here. The first test is viral test. So this kind of test, they detect the person has a current infection. So this will say if you're currently exposed to coronavirus or not. Okay, and this is actually done by using a swab test where you collect a sample from your respiratory system by using a swab test in your nose or your throat. And it is usually carried out in an instrument called RT-PCR, which I'll be discussing in the further slides. This is one type of test which is approved by FDA. Another type of tests are antibody tests. This test cannot detect a current infection. As the name itself indicates, antibody tests are looking for antibodies, right? So when you get exposed to an infection, your body takes at least 1 to 3 weeks to produce that particular antibody. So these antibody tests cannot detect current infection, rather they can detect a past infection. So once you get this test done, you would know whether you were previously exposed to this particular virus. And if so, do you have an immune response? So did you produce any antibodies for this virus? This is what you're going to look for in this type of test. Why is it essential? Because if you produce antibodies for this, for a particular antigen or a virus, it means that there is a very less probability, or in fact zero probability, that you might get affected by that virus again. So in order to figure that out, they're doing this antibody tests. So this is a serological test, that is, we collect the serum. It could be blood or plasma, and we continue the test. We use the human blood or plasma which has been exposed to this virus to actually do the analysis. This is done by ELISA, which we just discussed, and I'll show you how they actually apply to COVID-19. So let's see viral tests. Like I said, it is done by RT-PCR. RT-PCR stands for reverse transcriptase polymerase chain reaction. So this is the instrument which is used for testing the coronavirus and current infection. So it works on the principle of reverse transcription. So what you do is, when you have an exposed sample in your lab, the first thing you do is extract the RNA from the coronavirus. Once you extract the RNA, you generate the DNA by reverse transcription. So that hence the name reverse transcriptase. So from RNA, we generate the DNA by reverse transcription. And of course, you need a lot of factors for that, like DNTPs, magnesium ions, and other transcription factors to facilitate this reverse transcription. Once the DNA is produced, generated, we amplify the DNA by polymerase chain reaction. And based on the signal, you will figure out, whether or not the person is exposed to the virus. So this, like I already discussed, this will give you information about the current infection. This will tell you if you are currently exposed to coronavirus or not. The next type of tests are antibody tests, and of course ELISA is one assay which can do that, and ELISA stands for enzyme linked immunosorbent assay. So the first thing you do is, you take the ELISA plate, and you coat the well with the SARS-CoV-2 protein, which is the coronavirus protein. And then we take the exposed samples, the samples which come to our lab to be tested. These samples are incubated with the protein. So this is an antibody test, right? So if the antibodies are there in the sample, they will have to bind with the protein, right? So the antibodies bind to the protein, and they form an antibody protein complex which can be detected by using colorimetric or fluorimetric detection. So if there is a color change, it means there is an antibody protein complex formed, which shows that the antibodies are present in this particular sample. That means this person has been previously exposed to COVID-19, and he has a primary immune response, and he has produced antibodies for this virus. And there is a very little chance that this person might get exposed to the virus again. So this is how a typical ELISA works for coronavirus testing. And I would like to just briefly give a particular interesting fact here. CDC has initially developed a test which can detect just the SARS-CoV-2 virus in any exposed specimen. But recently, another test has been developed where you can simultaneously detect the influenza virus along with coronavirus. I think two types of influenza virus, virus A and B, can be detected along with coronavirus. I mean, this is a good breakthrough because you are detecting almost three types of virus simultaneously. So you are monitoring the coronavirus spread. At the same time, you're keeping a surveillance on influenza spread. Because you're doing simultaneously, you're saving a lot of materials, personal, because the biggest challenge right now is we are running of test supplies, right? So when you're doing simultaneously, you're going to save time, the materials and the personnel, and the speed is also increased, and you get the results three times faster. So I thought it's a very good interesting breakthrough, and it's an interesting fact to share.